term, you are going to learn the DNA, how the DNA was discovered, what is the structure of DNA, how the replication of DNA takes place, then what is the transcription and translation, what is the concept of transcription and translation, and you are going to learn certain mutation types of the mutations, okay, and then after you are going to learn the plasmids, types of the plasmids. This way your syllabus is designed and very fast we have to finish your syllabus because discuss about the DNA, how the DNA is discovered as the genetic material. All of you somewhat knowledge about the DNA, okay, and all of you learn very much about the DNA in and these chromosomes are made up of the nucleic acids and proteins. In 1928, the scientist Griffith discovered the experiment of the transformation, and right from the Griffith experiment, the breakthrough, this experiment takes place, a breakthrough in the science into the genetics specifically, and developments about the DNA, discoveries about the DNA are made after the Griffith experiment. In 1944, again, the three scientists, Avery, McLeod, and McCarthy, they discovered the DNA acts as the genetic material. Then in 1953, DNA acts as the genetic material into the bacteriophages also, into the viruses also. Then some scientists in 1955 and 56, they explained that or demonstrated that RNA also acts as the genetic material in certain RNA viruses. Those viruses which have the gene, which have the RNA as the genetic material. In 1953, the scientists Watson and Crick proposed the model of the DNA. Genotype means it is the genetic or genotype, it is the genetic makeup of the cell which contains all kinds of the information about the life of the cell. Uh, Here, that is we call it is the genotype means genetic makeup of the cell. We can say that genotype contains every and each information about the life of that cell. Cell, why I'm calling because every cell has the genome as the DNA. Okay, phenotype. That is the phenotype means the expression of the genotype. These characters are get expressed into the or these are called as the observable characters into that individual, into that cell. These observable characters are called as the phenotypic character. These characters may be the morphological characters or maybe the biochemical character. This is called as the phenotype of that cell or that organism. Okay? Then the nucleic acids are of two types, all of you know, DNA and RNA. Deoxyribonucleic acids and ribonucleic acid. Okay. First experiment was performed by the scientist Frederick Griffith in 1926, and he discovered the uh, transformation process into the Diplococcus pneumonitis bacteria. But after the Frederick Griffith, the first direct evidence about the DNA access genetic material was given by the Avery, McLeod, and McCarthy, these scientists in 1940. They demonstrated that Griffith, whatever the experiment performed about the transformation, that transformation takes place due to the DNA because the Griffith this experiment or phenomenon of the transformation was discovered by the Frederick Griffith into the 1928. This scientist took the pneumococci or Streptococcus pneumoniae as the experimental organism, and he uh, demonstrated that the Diplococcus pneumoniae exhibit the change in the information or change into the character, which is get expressed into the phenotype, okay, which is observed that the change in the character takes place due to the transformation. Okay, so that uh, he performed this experiment of the transformation. Now you understand about the this experiment carefully. So he selected the two phenotypic uh, mm, strains showing the different characters phenotypically. Okay, and he Griffith first selected these strains on the basis of these characteristics, the presence or absence of the surrounding capsule, or every bacteria in FY base, certain pathogenic bacteria have the ability to form the capsule around the cell, and that capsule, it is made up of the polysaccharide. Okay. Second character, he selected the type of capsule it is produced. Means in certain bacteria, this polysaccharide is present predominantly. In certain bacteria, this polysaccharide, different polysaccharide is present. In this way, this different different capsular material is present into the cells, even though the bacteria having the same species. Okay, we can call them, these are the strains. Okay, and hence he numbered these strains. He called this grown these cells which forms the capsule or which do not form, and he grown these cells onto the blood agar medium. The cells which produces the capsule, they forms the uh, slightly smooth colonies on the blood agar because of the secretion of this polysaccharide outside the cell. Because of these polysaccharide or capsular material, they produces certain soft colonies like this. Okay, and these colonies are shown here. Okay, these colonies are soft or larger because of the production of the capsule. You can see here the in second strain, these colonies are rough colonies because they smoothness in these colonies are not there, because they do not secrete the capsule. So that on this basis, the smooth strain and rough strain, he termed as the Streptococcus pneumoniae smooth strain and Streptococcus pneumoniae rough strain in this way. Okay. Then rough, you can see here rough colonies and the smooth colony. One more important character of this strain is that this uh, smooth cells or the smooth variety of the Streptococcus pneumoniae, it is the pathogenic for the human beings or for the animals. And rough colonies, the cells which do not form the capsule, these cells are non-pathogenic to the human beings. Okay, hence this is the differentiation. Now you remember the smooth variety of smooth bacteria, that is the Streptococcus pneumoniae, which forms the capsule, which forms the smooth colony. The another strain of the bacteria, that is Streptococcus pneumoniae, which is the which do not form the capsule, and it is the rough. They forms the rough colonies because of this character, and these are non-pathogenic. Okay? So here In this experiment, he further categorized this capsular material. He given the name numbers into the Roman, okay, first smooth type of the cell, second smooth type of the cell, third smooth type of the cell, okay. And from this smooth type variety, it is the pathogenic, okay. And rough type variety, it is the non-pathogenic. Third smooth type variety is selected. First smooth, second smooth, and third. And he selected for his experiment third smooth cells. Then he selected another variety, that is the non-pathogenic rough cells or rough second type, second rough cells, like this. This is not, but these rough cells lose the ability to synthesize the capsule due to the mutation. Okay. This is a mutation behind the uh, formation of the capsule, because certain mutation into the gene which synthesizes the capsular material, and because of the mutation they unable to synthesize the capsule. And hence what did this Griffith, you can see here the he took the living cells, okay. Type third smooth cells he took. First, he took the type third smooth cells, okay, like this, type third smooth cells. And then after that, he injected this smooth cell. Smooth cell causes the pneumonia into the mouse or animals, you can say that. And because of that, it death of the mouse take, like this. Okay, it was the initial experiment performed onto the mouse. Okay, he took the pathogenic variety, that is the third smooth cells, and injected into the mouse, so that mouse died. Okay, mouse died, and after that, from the body of the mouse, type third smooth cells are recovered, because these are injected, these grown into the mouse, these are caused the disease into the mouse, and because of that, death of the mouse takes place. And from this mouse, he recovered the live type third cells. Then into the second trial, he took, okay, living type second R cells, okay, and he injected into the mouse. Mouse survives here because these are non-pathogenic. They cannot form the capsule. These are non-pathogenic. Hence, the mouse survives. And from this, no living bacteria were isolated from the mouse, which belongs to the live type third cells, like that. Okay. In third trial, what he did, third trial, he took the heat killed type third cells. Now, so in first trial, what he did, he took the living type third smooth cells, which are the pathogenic. And in third trial, what he did, even though the cells are pathogenic, but he heat killed. Because of the heat killing, dead cells cannot cause the disease. And hence, these cells, after injected into the mouse survives there. Mouse survives. And because of that, there is a no death of the mouse, and there are also no growth of the cells, bacteria from the body fluid of the mouse, because these are the dead cells. But what he did into the fourth trial, okay, in the trial 4, he did, he took the rough type second cells, he took the smooth type third cells. Okay, means that you can see here living type second rough cells. These are non-pathogenic. They does not cause the disease. No death of the mouse. Heat kills type second, type third smooth cells. These are heat kill. These are dead cells. These are injected previously into the previous trial. No death of the mouse. No sign of the disease. No death of the mouse. And he mixed these cells together. That is live type R second cells and heat killed type third smooth cells. And what would be the result after injecting into the mouse? Mouse should survive or mouse would have been survived. But here you can see the mouse, death of the mouse takes place. And from the body fluid of the mouse, he also recovered the type third live smooth cell. Okay, and because of that, he concluded that there is a conversion of type second rough cells into the type third smooth cell, isn't it? Because of that, he concluded that there is conversion of the type second rough cells into the type third smooth cells. The Griffith unexpectedly discovered that because he initially tried separately when he injected the type second rough cells, no death because these are non-pathogenic cells, no death of the mouse. He injected separately the type third smooth cells, no death of the mouse because these cells were heat killed. And in final trial, what he did, he took the heat kill smooth cells as well plus live rough non-pathogenic rough cells and injected into the mouse and mouse shown the signs and symptoms of the disease, okay, that is pneumonia and death of the mouse takes place. And from the body of the mouse, type third smooth cells were recovered and he it was unexpected result. Otherwise, mouse should have the survive. Okay, it is unexpected result, and because of that, he got in puzzled, and he thought that there might be the transformation of the type second rough cells to the that is the type second rough cells non-virulent to the type third virulent cell. Okay, and then thus transformation of non-virulent type second cells to the virulent type third cells means virulent means pathogenic. It is a transformation of non-pathogenic type second rough cells to the no, pathogenic type third cells uh, takes place. Okay, but he cannot explain that how this transformation took place, how the rough cells or how the non-pathogenic cells are get converted into pathogenic cells. But he described this phenomenon as the transformation.